Hello, and welcome back to the Sound of Science. Today, we're going to be talking about evidence for evolution. There are four types of evidence that we can talk about: fossil records, DNA, comparative anatomy, and embryology. And I'm going to go through each type as we move through this lesson. Okay. Before we even really talk about the fossil record evidence, just some information that you need to know. The Earth's been around for 4.5 billion years. And it takes us a whole billion years after the formation of the Earth before we start to actually see life. Um, Fossils can only form in sedimentary rock. And because we don't have sedimentary rock everywhere, the fossil record actually ends up being incomplete. There's gaps. There are places where we suspect we should be seeing fossils that we're not. So those sorts of things mean that the fossil record is, while it's great evidence, it's not the only evidence that we're going to rely on because it's incomplete. So when we talk geological time scale, this is huge. And as you can see here, it's going to go back billions and millions and so on. As we move through, you can see that we have what we call eons, eras, and periods. These are all just large periods of time, and it's based on what we're seeing. It's not like each eon is a billion years. It's really based on what's happening and what we can see. So if you take a look at some of the key events, earliest life shows up there at the bottom. Invertebrates start to become common a little more than 500 million years ago. The Permian mass extinction, which takes place during the Permian, right before the Triassic, you can see that there. Extinction of the dinosaurs is occurring in the Cretaceous. This is all truly, as you get towards humans, I mean, that's really fairly recent in geological time. And so keep that in mind when we talk about evolution. We're not talking about a short period of time. We are talking about generations upon generations. It's not something that can happen quickly. So where do we come into all of this? If you look at this, this is saying that 4.6 billion years is occurring in the course of 1 hour, because we really can't reference 4.6 billion years. That's a lot of time. We don't have any reference for that. But we do know how long an hour is. And so if we're saying that at the very, very beginning, in the first minute, Earth's crust forms, it is not until 59 minutes, 59 seconds, and 9/10 of a second that humans even appear, or that modern humans appear on Earth. And really, for us, that's about 100,000 years ago or so that we start to see humans. So, just to kind of give you a reference of how long life's been on the planet without us, it's quite a period of time. Now, let's talk fossils. So there's a couple of different types of fossils that we can talk about, and it can be fossils that are traces or body fossils. Some are going to be classified as preserved remains. These are the ones that we're going to see appearing in ice or amber or tar, and this is where like the whole body's been preserved or a portion of the body's been preserved. Or we have mineralized fossils. This is where the traces or the fossils themselves are going to become stone. They're actually turned into rock. Two ways that that can happen. The first way is probably the way that most of us think of, and this is the mold method. So this is where you have, like for instance, what you see over on the left-hand side here, where the dinosaur dies and it gets buried in sediment and it starts to decay, but it leaves behind that hollow space where its bones were. And that hollow space is actually going to fill up with minerals, much like a cave is formed, where the minerals sort of drip into place and they're brought in by water and they slowly form formations. The same thing happens with fossils. When fossils are forming, we are getting minerals building up in the hollow spaces left behind in the sedimentary rock. Um, We also have a mineralization process that can occur where the calcium in the bones actually swaps places with minerals. Um, and therefore, they just literally turn into stone or turn into rock because the minerals are essentially replacing segments of the bone. Regardless of how it forms, fossils are always going to be found in sedimentary rock. They're always going to be um, some sort of trace that's left behind. It may be the mold that was left behind by bones as they decayed, or it could be footprints. Something as simple as a footprint can be left behind and form a fossil. So, fossils give us lots of information, not just how the organism looked or how the organism walked in the case of footprints or what was left behind, but it actually can also give us a date, a time period that we're looking at. And there's a couple of ways that we can date fossils. To determine their age, we have either relative dating, which is basically determining the age of the fossil based on the layer of rock. And relative dating means it's relative to what's around it. So it's going to be older than some layers or younger than other layers. We also have radiometric dating. This is based on the ratio of radioactive isotopes. Carbon 14 is one of the most common, and I'll explain how that works here in just a minute. Important to remember, an index fossil is any fossil where we are certain of its age. We know how old this fossil is. We know when this organism was alive, or we know when something had to be left behind. And we can actually use that to determine the age of the fossils that appear in the same layer with it. Essentially, relative dating really just means that the oldest layers of rock are going to be found on the bottom. So if you look at this picture that I pulled, just so you can sort of see what we're finding here, um, the law of superposition says that those in layer A have to be older than those in layer B. And layer B is older than layer C, and layer C is older than D, and so on and so forth. And so if you look at this, you can actually see where certain organisms may have appeared, or what organisms were there at the same time. For instance, in layer A, we have trilobites and we have ammonites. These are both actually really common fossils that have been left behind. And we know that these guys were contemporary, but we also know that ammonites lived longer than trilobites. We actually had them for a lot longer, and they were also contemporary with certain types of bivalves and fish. And so we actually can see how they extend into the next layer. All of this, like I said, is based on the law of superposition. Law of superposition is lengthily worded, but really what you guys need to know is it just means that layers that appear on the bottom have to be older than the layers that are on top of them, which makes sense. It also actually deals with layers that occur through. So sometimes you see cutouts. If you look at layer V, it's actually cutting into layer W, X, and Y. And that means layer V had to occur after W, X, and Y. It's an intrusion. And it can occur in those layers unless those layers are already there. So essentially, we're just using stratigraphy to determine age. Let's talk about radiometric dating. There are radioactive isotopes in all living things. Well, over time, those radioactive isotopes are going to decay into more stable isotopes. For instance, carbon 14 decays into the more stable form of carbon, which is carbon 12. It just basically is going to lose a couple of neutrons. That's all that's really happening. The ratio of the radioactive isotope to the stable isotope actually tells us how long it's been since the organism died. So we indicate this by using a term called half-life. Half-life is how long it takes for half of the sample to decay, and it's really half of the remaining sample. So in carbon 14 dating, it's 5,730 years. That's how long it takes for half of the sample of carbon to go from carbon 14 to carbon 12. So if I look at a fossil or look at remains that have been left behind, and half of the carbon 14 and half is carbon 12, then I know that it's been at least 5,730 years, give or take about 25 years. If I look at it though, and it's 25% carbon 14 and 75% carbon 12, then that means that two half lives have passed. The first half life meant half of it decayed. The second half life meant half of that was remaining, that 50% has decayed. Once that quarter of carbon 14 remains, then we're looking to see half of the half to decay to determine that the next half life has passed. And each time half of the sample that remains decays, another 5,730 years will have passed. Here's a very, very simple example. Sample has 12.5 radioactive carbon isotopes and 87.5 stable carbon isotopes. How many half-lives have passed? And so you're going to make the assumption, of course, that it's 5,730 years per half-life. But if you look at it, 87.5. So the first half-life would have taken us to 50. The second half-life would have taken us to 75. The third half-life actually takes us to 87.5 if we just take half of whatever remains each time. So that means we've passed 3 half-lives. If we've had 3 half-lives, then that means we would take 5,730, multiply by 3, and that's going to give us our answer. As you can see here, 3 half-lives, 17,190 years have passed since this organism was last alive. I know, radiometric dating is one of those things that if you just understand the concept, usually you'll be okay. Now let's talk about comparative anatomy. This is the second type of evidence for evolution. And to start, we're going to talk about homologous structures. So homologous structures just means that these are structures that are similar. They come from the same embryonic tissues. They've been inherited by organisms that have a common ancestor, but they're probably going to be used differently. Usually with comparative anatomy and homologous structures, what we're really talking about is structures that we know were shared by a common ancestor, which is why all the different organisms have that structure, but they're often used differently. The example that we're giving here is our human arm. Cat legs, whale flippers, and bat wings are all used differently, but they all actually evolve from the same common ancestor. Now, this is not to say humans have evolved from bats or whales or cats. It just means that humans, cats, whales, and bats all shared an ancestor at some point. And it could have been 3 billion years ago or 2 billion years ago. But at some point, they all shared a common ancestor. And the more similar the structures are, probably the less amount of time that's passed since they had a common ancestor. With humans, when we start comparing bones, one of the really cool things that I like to point out is that we have a pattern of bones. So we have this humerus, right? Radius and ulna, carpals, metacarpals, and phalanges. This is a pattern of one bone, two bones, lots of bones, digits. That's what we see in human arms. If we look at the cat bone structure, one bone, two bones, lots of little bones, digits. Whale flipper, one bone, two bones, lots of little bones, digits. And again, let's take a look at the bat wing. One bone, two bones, there are two bones there, lots of little bones, and then digits. And that's what we see in all these organisms. Even though these structures are being used differently, these structures are all coming from a common ancestor that had the one bone, two bones, lots of little bones, and digits structure with them. Along with homologous structures, we actually also have analogous structures and vestigial structures. So vestigial structures are going to be structures that we don't use anymore. For instance, dolphins and whales have hip bones, they have pelvic bones. Even though they don't have limbs there, they will have these little itty bitty bones that just sort of hang out in their blubber and they're just there. Sometimes we actually see little vestigial legs on snakes as well, these little tiny bone structures. And these are things that are just not being used anymore. And they're there because one of their ancestors needed them. And the structures over time have just gotten smaller. Analogous structures are going to have similar functions, but they're not the result of a common ancestor. They are just may have similarities because they have evolved for the same purpose. So analogous structures have similar functions or similar purposes. So the bird bones actually have that similar structure, the one bone, two bones, little bones and digits, whereas a butterfly wing is going to be very different. Actually, it doesn't have bones at all because it's from an invertebrate, but it's going to be an extension of the exoskeleton. So they don't have much in common except that they're both used for flight. So molecular evidence. Molecular evidence actually deals with DNA evidence. So what we might do in this case is look at a sequence for a particular protein and look at the amino acid sequence and then determine how many amino acids are different versus what was there. Because here's the thing, guys. The whole thing, I don't know if you guys have heard it or not, but it's like, hey, we share 50% of our DNA with bananas. That's actually true. We do, because there's a lot of things that we have that all living things have. And so we share some DNA sequences with organisms that we're not particularly closely related to. The more closely related we are, the less difference you're going to find in the DNA between that organism and the other that you're comparing it to. What we see here are humans, monkeys, a specific type of monkey, a mouse, chickens, frogs, and lampreys. And as you go down, if we're looking at amino acids in a sequence for a particular type of protein, in this case, we're looking at hemoglobin. The total protein or a polypeptide chain length is 146 amino acids. 146 amino acids all in a row, and we're comparing them. So obviously humans is the same as humans. Rhesus monkey only has 8 amino acids that are different from the human molecular chain. That's pretty closely related. Mice are only going to have 27 differences. Chickens, we're getting a little further out, 45 differences. Frogs will have 67, and lampreys will have 125. So the higher number of protein differences or differences in amino acid sequences in specific proteins gives us an idea of how long it's been since we shared a common ancestor or how closely related we are. Those that are more closely related, we're going to be seeing less differences. But the molecular evidence is actually really accurate. We can get a really good picture of how different organisms are closely related. Sometimes you actually will find that an organism or two organisms won't have any differences in their amino acid chain for a certain protein. And if that happens, then what you do is you actually look at a different protein. So you compare more than one protein as you're building up the evidence you have. Fourth type of evidence. You guys have done a great job hanging in. Embryological evidence or developmental evidence. All this is looking at is organisms as they're developing as embryos and how closely related they are, what characteristics they share, what characteristics they don't share, to help us determine how closely related different species might be. Here we have a fish, salamander, tortoise, chick, hog, or pig, calf, rabbit, and human. And if you look at the very top row, they all look really similar. And in fact, if I was looking at the top row only, I wouldn't be able to pick out the human from the salamander. As we move to the second row down, we see that, like, for instance, the fish and the salamander are already very differentiated. whereas the human and the rabbit, the calf and the hog are all still pretty similar, and the chicken and the tortoise are fairly similar. And so we can actually see that humans are more closely related to the other mammals than they are going to be to the salamander or the fish. And then, of course, as we get to the row 3, we can see things that we still have in common or things that have changed. And this gives us an idea of how long it's been since humans shared a common ancestor with each of these types of species. So that's your 4 types of evidence. Make sure you understand each type of evidence. Make sure that you have written down what each type of evidence is and how it gives us information. So again, that's all we have time for today, but I'll see you next time on The Sound of Science.